Hello children, how are you? Hope you are enjoying my video classes. So, today almost we have reached towards the end of second topic of NCERT textbook, that is sexual reproduction in flowering plant. During these days we are talking to you about the different events of sexual reproduction and the post-fertilizational event. Today we will be discussing about the embryonic development. In previous class, in continuation with that, the dicotyledonous and monocotyledonous embryo development, what are their structures. So let me talk to you first of all the embryo of a dicotyledon. This is the dicotyledonous embryo development, but still it is not completely mature. When it will completely mature, what will be the structure of that dicotyledonous embryo? Let us see. Let us draw a diagram, simplified diagram I will draw you for, so that you can also easily draw in your examination. So by name itself, you can think about the dicotyledonous means having two cotyledons, So this is one, this is the second one, this is the embryonal axis. Embryonal axis, slight modification. This is the embryonal axis. This is the root cap, So this is the part embryonal axis which terminates at one end with two cotyledons. These are the two cotyledons. These are the two cotyledons. If I little modify, then it will be easy for you to get a proper clarification. This embryonal axis, this part above the level of cotyledon, the part or portion of the embryonal axis above the level of cotyledon, what you call as epicotyle, which is having an extreme area. Epicotyle give rise to plumule, and the embryonal axis below the level of epicotyle called hypocotyle. This hypocotyle terminates into radicle, otherwise called root tip. Radicle or root tip, which is covered by root cap. So this is a very simplified diagram of dicotyledonous embryo having an embryonal axis. This embryonal axis terminates with the two cotyledons, that means there are two cotyledons, hence it is called the embryo of dicotyledon. Now the level or the area just above the two cotyledon, the embryonal axis which is having area above the two cotyledon is called epicotyle, which gives rise to plumule, and below the level of origin of epicotyle, that is the hypocotyle, which gives rise to radicle or root cap, which is covered radicle or root tip, and this is covered by root cap. This is the embryo of a dicotyledon embryo. Now, if I discuss about, let me rub this part. If we go and have a view over the monocotyledonous embryo, especially in case of grasses. In case of grasses, all the monocotyledonous embryo, monocot itself suggests that it is having a single cotyledon. If you see the embryo of grass, which is also belonging to monocotyledon, now if you see the structure, it is a little difficult, but with your little more effort you can easily draw the diagram. And many times in your examination, including board examination CBSE, you will be getting this types of monocotyledonous embryo structure. So you have to be very acquainted with a simplified diagram I am drawing for you. So this is the embryo of a monocotyledon. See how you can easily draw this diagram with less effort. This terminates in this way. Now this is the embryonal axis, the embryonal axis. The embryonal axis. One side it is extended, this is the embryonal axis. This embryonal axis at one end of this embryonal axis there is an attachment, a lateral appendage like structure. This is called as scutellum. Scutellum. This is called scutellum. If I shade this area of scutellum along with the whole embryonal axis, if I shade this area, so a single cotyledon appears as a lateral appendage at one side, what we call as scutellum, and the tip of embryonal axis just above the level of origin of scutellum is the epicotyle, and this is the shoot apex. shoot. This is the floral or leaf primordia. Leaf primordia. This is the leaf primordia, which will give rise to leaf. Leaf primordia, which will give rise to leaf. And it is covered, the epicotyle covered by a thin foliage of structure or covering or sheath called as coleoptile. 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 The portion of embryonal axis, below the attachment of the cotyledon. This is the cotyledon covered by scutellum. Below that attachment of, this is the attachment, the portion below the point of attachment of this cotyledon, this area of embryonal axis, what we call as hypocotyle, hypocotyle, which give rise to root tip, otherwise what you call radicle, which is covered by, if I make a slight modification for your easy understanding, so this is the radicle, and radicle is covered by root cap. So this is the root cap. So you can compare the embryo of dicot and embryo of monocot. This is the dicot embryo, and this is the monocot embryo. So this is the post-fertilizational changes. That means which part will develop into this embryo? It is the zygote will develop into embryo. And what will be the fate of the primary endosperm cell? It will convert into endosperm. Endosperm, after its formation, after certain amount of endosperm is formed within the seed, then only the embryo maturation, embryo development takes place. So that during the development of embryo, it can be ensured that it is getting sufficient amount of food material from the endosperm. Is that very clear to you? Now, what will be the post-fertilizational changes for the entire ovary and the ovule present within it? So the post-fertilizational changes, if I make a change here, if I rub this part, so after fertilization, the ovary changes into a structure called fruit, and the ovule present inside, changes into seed. If I again try to make you remember that the structure of a pistil which contain ovule, ovule is surrounded by two layers of integument. You might surprise why I am drawing this diagram again, because something more I shall discuss here which will require this diagram again. So this egg after fertilization getting converted into embryo and this polar nuclei getting converted into endosperm. Now the entire ovary, this part, ovary, this ovary after fertilization getting converted into, this is the ovary, this part getting converted into fruit. And this ovule means this part only, including the integument. This ovule getting converted into seed. Now if I take a seed, what are the structures? What are the content present in the seed? So structure of the seed. Structure of seed. Very simple way, if you take out a mango seed, it will have, as it is dicotyledon, it is having two cotyledons like this, having an embryonal axis. This is the embryonal axis. This will give rise to a plumule, radicle. These are the cotyledons. And there is a seed cord. This is the seed cord. Seed cord. Seed cord. Now, if I simply draw a diagram of an entire seed, you will see the two cotyledons, as I have told, as I have drawn the diagram also. These are the two cotyledons. These are the two cotyledons, the embryonal axis. Now it is covered by seed cord. Where from this seed cord came? It is this integument, as I told in my previous classes also, that the ovule is surrounded by one or two integument. It is this integument is forming the seed cord. And as I told you, here also you can see the seed cord is not covering the ovule entirely. At one end there is an opening called micropyle. This is the micropyle. So it is this opening, small notch-like or scar-like structure present at one tip of the seed. That is the micropyle actually of the seed cord. What is the function of this opening or aperture? It is through this aperture, small notch-like aperture. It is through this aperture present in the seed covering or seed cord, oxygen and water enters into seed. This is what is very much required for seed germination. If you keep the seed in isolated place, in a dehydrated form, the water content will reduce from 10 to 15% only. this will not help or promote the seed germination. In that case, the seed will remain in dormant condition. When again this moisture become more, oxygen is getting, then it can facilitate for the germination, and through this the oxygen and air will, the oxygen and water will enter into the seed. Is that very clear to you? Now, the seed can contain some amount of, the seed can contain some amount of seed, can contain some amount of reserved food material in the form of endosperm within it. If it is the case, then it is called as albuminous seed. Albuminous seed. Where on course of development, some amount of, maybe little, maybe more, reserved food material, that is endosperm, is still present within the seed. That type of seed we call albuminous seed. Whereas in other cases, can you give an example of albuminous seed? For example, paddy or rice, wheat, where the seed contains sufficient amount of endosperm in it. On the other hand, if you see the groundnut or say leguminous family, any seed, the seed do not have any reserved food material after its maturity or during its maturation. The food material in the form of endosperm has already been consumed, remaining behind the two cotyledons and the embryonal axis. That type of seed we call non-albuminous seed. Example, say groundnut or say pea. So is it clear to you? Albuminous seed, non-albuminous seed. Now, post-fertilization change, as I told you, the ovary changes into fruit. So if I draw a simple fruit, a sectional view, say suppose in case of mango, a sectional view. So this is the seed, having two cotyledons with the embryonal axis, two cotyledons, and covered by sheath coat, Now, the fruit is this part is the modified, it is the modified ovary. Now, during the formation of fruit, it is the ovary which is getting modified into fruit, whereas other part, style stigma and the thalamus part gets degenerated. But in certain case, so what is the part which converts into fruit? Only the ovary. This types of fruit we call true fruit. True fruit. So what is the true fruit? True fruit is a type of fruit wherein the fruit is formed only from the ovary. Only from the ovary. On the other hand, if you take an example of, say, apple or strawberry, here if you make a cross section of apple, cross section. This inside view you are getting like this. Wherein apart from the ovary, thalamus part, this is also taking part in the fruit formation. This types of fruit, apple, what we call as false fruit. It is mango. So why it is called false fruit? Because of not only the ovary but the thalamus part is also taking part in fruit formation. Now whatever be the case, the apple or mango, the fruit is the post-fertilized ovary, irrespective of the type of fruit. In case of juicy fruit, the outer covering of fruit, what we call as the outer covering, rather to say the covering or the ovule, outside the ovule, the ovary which is covered. So this outer covering, what we call as pericarp. Pericarp. This pericarp is having three parts. The outermost one, that is this one, the outermost one, here also, the outermost one, which is forming the fruit pill. This pill part is called as epicarp. Epicarp. This part, juicy part or eatable one, called mesocarp. And the seed covering coat outside the seed, this one, it is the endocarp. Endocarp. So the pericarp is having three parts. One is epicarp, second is mesocarp, and the third is endocarp. Is that clear to you? Endocarp covering the seed. Endocarp covering the seed. This is the endocarp here. Here it is the endocarp. Endocarp covering the seed. This is the seed, apple seed, apple seed, apple seed. Is it very clear? Now, in most of the cases or almost all the cases, the fruit is formed after fertilization where the ovary changes into fruit. But there are cases where ovary without fertilization changes into fruit. That type of fruit we call parthenocarpic fruit. Parthenocarpic fruit. What is the term I have used? Parthenocarpic fruit. Parthenocarpic fruit. That is where the fruit is formed without fertilization. And hence there is no question of fertilization here. So there is no question of seed formation because seed is the post-fertilized ovule. So there is no question of seed formation. So these types of fruits are seedless fruit. So remember, parthenocarpic fruits are the seedless fruit. Seedless fruit. Seedless fruit. Say for example, very common example, banana. Banana is a parthenocarpic fruit. It is developed by using phytohormones. Is that clear to you? But I will draw your attention here towards a very specific and peculiarities at the post-fertilization event. The two terms we will use, one we call apomixis and the other is called polyembryony. So let me rub this part so that I can discuss detail about polyembryony. Number one, apomixis, and number two, polyembryony. Polyembryony. What is apomixis? Apomixis is a condition. Generally, as we by that time so many times told that seed is the post-fertilized ovule. But some cases, for example, grasses, best example, where the seed is formed without fertilization. If fruit is formed without fertilization, that term we have used, parthenocarpic fruit. If seed is formed without fertilization, that term we call apomixis. And what is polyembryony? Generally in angiosperm, as we have so many times talking about angiosperm, and there we told you that in angiosperm the embryo sac development is called monosporic development. Why? Because within the, so again I have to draw the diagram so that you can get a very clear concept. Diagram of again pistil. So this polyembryony term you have to be very, very clear. This ovule contain nucellar cells full of reserve food material. In early classes we have discussed. Now these are the nucellar cells forming the nucellar tissue. It is covered by two integument, one or two integument. Here I am showing two integument. Now during the time of embryo sac formation, that is towards the micropylar end, one of such nucellar tissue become highly developed. One of such nucellar tissue towards the micropylar end become highly developed and converted into embryo sac. Is that clear to you? Around the embryo sac, now what is there? If I draw a bigger embryo sac, so this is the embryo sac. Now this embryo sac developed within the ovule. This is the embryo sac. Now within the ovule, around the embryo sac, the tissues are still present, the nucellar tissues. Now, normally, these nucellar tissue, after maturation of the embryo sac, gets degenerated. But in certain cases, some of the nucellular tissue protrude inside the embryo sac, and they also form small embryo sac. Suppose this nucellular tissue protrude inside, forming one embryo sac. This is forming one embryo sac. This nucellular tissue protrude inside, forming another embryo sac. So if I rub this part, so the nucellular tissue protruding inside the main embryo sac and forming small embryo sac. So this bigger embryo sac ultimately getting, after fertilization from zygote, getting converted into embryo, and this small one also getting converted into embryo. So within the ovule now many embryos are there. This phenomenon we call polyembryony. So presence of it is the presence of more than one embryo within the seed is called polyembryony. Example is citrus fruit, lemon. So today we have discussed in detail about the structure of monocot and dicot embryo. Especially I have given a lot of emphasis on the dicot embryo because it is little difficult, but with little effort and practice you can draw the diagram. It is having the single cotyledon extend at one side. So this is the structure having a embryonal axis, the upper part of the embryonal axis from the origin of the cotyledon. Above the origin layer, that is epicotyle, and below the layer, hypocotyle. Epicotyle gives rise to pseudapex, the leaf primordia. The epicotyle is covered by a thin foliage structure called coleoptile. The hypocotyle is covered by a thin foliage structure called coleorhiza. Coleorhiza I have written somewhere. This structure is called, again I am writing this structure, this one. So let me write here. This one, this coverage is called coleorhiza. So this is the structure of monocot embryo. Dicot embryo comparatively simple, having two cotyledon and embryonal axis. The level of embryonal axis above the origin of cotyledon, epicotyle, which gives rise to plumule. Plumule is the future shoot. The embryonal axis below the level of origin of two cotyledon is called hypocotyle. Hypocotyle terminates to root apex, otherwise called root tip, or commonly called radicle, which is covered by root cap. And we have discussed about the structure of seed and the seed coat, the fruit, the parthenocarpic fruit, where the fruit is developed without fertilization, and the apomixis, the phenomenon where the seed is developed without fertilization, and the polyembryony, detail how it is formed, I have discussed. So I hope the thing is very clear to you. This is how we have completed the sexual reproduction in flowering plant, the different structures, pre-fertilization structures and post-fertilization event and structure. I am sure the thing will be very much clear to you once you go through deep and the little effort you require to draw the diagrams. With this, thank you for patiently attending my class. Next chapter we will be discussing in next day. Till that time, bye bye.